Hello everyone, Sangeeta here. Let's have a revision on immunoglobulin structure and functions. So from this portion usually the question has been asked are explain the structure and functions of immunoglobulin number 1 and number 2 differentiate between immunoglobulin A that is IgA and IgM or IgG and IgA. Sometimes it is amongst these four like any kind of differentiation can be asked. Well, let's go through with the answer. So, for the first question was explain the structure of immunoglobulin and functions of immunoglobulin. First of all, you should write the definition for immunoglobulin. Immunoglobulins are a Y-shaped glycoprotein produced mainly by plasma B cells that is used by the immune system to recognize a unique molecule, a harmful agent called antigen. That is a definition of immunoglobulin. These are the gamma globulins synthesized by the plasma B cells. Let's go in detail about the structure of immunoglobulin. If you see the structure of immunoglobulin, you can appreciate it as a Y-shaped molecule, isn't it? So, immunoglobulin molecule chains are connected by we will first of all we will go through with the figure in detail. We can see that it is made up of 1, 2, 3 and 4 polypeptide chains. Polypeptide chains, among this 2 is very heavy or long chain and 2 are very small or light chain. So it is made up of 2 heavy chains and 2 light chains. And you can appreciate multiple sites there. You can see that the amino terminal region, or you can appreciate 2 amino terminal region and 1 carboxy terminal region. And so many other things also labeled VH, that is representing variable heavy region, VL, that is representing variable light chain, then constant heavy chain and constant light chain. So the first thing is that you are supposed to draw this figure properly with proper labeling. You can even use various color for heavy chain and light chain and for the variable region and constant region. Okay, and you can see that it is joined together by interchain and intrachain sulfide bridges. And the neck of this immunoglobulin is known as hinge region. Why this hinge region? Hinge region is responsible for the flexibility of immunoglobulin so that this Y-shaped antibody or immunoglobulin can move towards the antigen and can go ahead with the reaction. Okay, let's see detail. All immunoglobulins have the same basic structure as a Y-shaped basic structure. It is made up of 4 polypeptide chains, 2 heavy H for heavy chains and 2 L chains. The 4 chains which is linked by disulfide bonds, and in previous figure we have seen that it has 2 light chain and 2 heavy chain. So the light chains can be either 2 types, a kappa chain or lambda chain. In an individual immunoglobulin, it may be usually a 2 kappa or 2 lambda light chains. It cannot be a kappa chain or lambda chain together. And the heavy chains are of 5 types, and usually it is designated by Greek letters alpha, gamma, delta, mu and epsilon. Based on these heavy chains, immunoglobulins are classified into mainly 5 types: IgA, IgG, IgD, IgM and IgE. In theoretical class, we have given a short form for this classification, GAMED, GAMED, that is representing all these 5 classes. The light chains and heavy chains are subdivided into variable and constant region. What is this name variable and constant region? Because the amino acid sequence of variable region is varied with various immunoglobulin. The amino acid sequence of constant region with variable with various immunoglobulins are constant in different immunoglobulin. That is why the name is given as variable region and constant region. So each light chain and constant region consists of variable region and constant region. So the L chain consists of one variable, that is VL, and constant chain consists of one variable region, that is CL. Then same like heavy chain, VH, and VH is there, only one variable region is there for heavy chain, but it has three constant region in the heavy chain, that is represented CH1, CH2, CH3. You can go through with the figure and understand. And each immunoglobulin has a hinge region, I said, that is responsible for the flexibility so that it can fit with the antigen surface. And the variable regions of both light and heavy chains form antigen binding site. And another important point, papain, an enzyme present in the papaya, which can digest or splits the immunoglobulin molecule into two fragments called FAB, that is fragment for antigen binding, and FC, that is crystallizable fragment or fragment for complement binding. So this is all about the structure in detail for immunoglobulin. So immunoglobulin, remember, basically it has a Y-shaped structure which is known as a monomer. IgG, IgE and IgD is a monomer which consists of only one basic unit, whereas IgM is a pentamer which consists of 5 such kind of monomers, and IgA is a dimer which consists of 2 such kind of monomers. Now the functions of immunoglobulins. The primary functions of immunoglobulin or antibody is to protect against the infectious agents or any foreign substances which enters into the body. Additionally, it has some other function, like it has some free radical scavenging activity or antimicrobicidal activity. And each class of immunoglobulins, we are going to see that each class of immunoglobulin has its own specific functions also. So this is the answer you are supposed to write along with the question as structure and functions of immunoglobulin. Now the second question is differentiate between two types of immunoglobulin. So already we have discussed it consists of 5 classes based on the heavy chain: IgG, IgA, IgM, IgT, IgE. Usually IgD having less importance, so it may not come. But the question may like differentiate between any these 4 types, between IgG and IgA, between IgM and IgE, or IgG and IgM, like that. For that, each class you should know thoroughly. Okay, let's have a look. IgG and IgA, if you are considering these two, IgG heavy chain is gamma and IgA heavy chain is alpha. IgG is a small monomeric molecule with two antigen bindings, right? Where IgA is a dimer, consists of two monomers unit joined together with a J chain, J for joining chain. Then IgG is the major class of immunoglobulin. It accounts for the 70% of total serum immunoglobulin, whereas IgA is the second most abundant class consisting 20% of serum immunoglobulin. Remember, it is a dimer, IgG is a monomer. Then IgG, which is the only antibody, can crosses the placenta and can provide the immunity to the fetus. IgA, which is found in external secretions such as colostrum, saliva, tears, respiratory, intestinal, genital, and genital tract secretions. So, the function is IgG is produced mainly in the secondary response and provides defense against bacteria and viruses, and also it gives immunity to the fetus. That is a function of IgG. IgA, it prevents attachment of microorganisms like bacteria and viruses to the mucous surfaces and helps to protect mucous membranes, mucous surfaces from antigenic attack and prevent aggressive foreign substances to the circulation. Now, differentiate between IgM and IgE. M is a pentamer, E is a monomer. Pentamer is joined together by disulfide link bridges. IgE, it is also known as reagin, and it is present very trace amount in human body, that is 0.004% only. IgM is the first antibody to be produced in response to antigen and is important in defense against bacteria and viruses. Whereas IgE is mainly responsible for the allergic activity. During allergic condition, the level is very much increased in persons. IgM is present on the surface of B lymphocytes and are monomers where it functions as an antigen binding receptor. IgE, which is responsible for the anaphylactic or immediate type of hypersensitivity and allergy, and it is mediated by release of histamine. So so that the allergic reactions occurs during because of the presence of this IgE. IgE also participate in defense against parasitic infections like helminthitis because of the worms. So this figure which explains the overall structure, functions and characteristic features of all these types of immunoglobulin. You can have a look and study in detail. Thank you.